Okay, students, is the sound and audio are clear? Okay, good evening. So, as we have discussed yesterday about the fertilization, how the fertilization takes place and how the development of female gametophyte takes place. Then we have seen for what are the different types of pollination are there, what are the different agents and what are their features. Okay, and then we have gone for pollen pistil interaction and how the pollen tube germinates and it enters into the micropylar end and it fertilizes the both the nucleus present in the ovum and our primary endosperm nucleus. Okay, and they are converted into zygote and sorry, polar nucleus, and they are converted into zygote and primary endosperm nucleus respectively. Okay, so after that, this today we are going to discuss about the topics or the processes or phases that are taking place after fertilizations. So all these procedures or all the developments of this embryo or development of this zygote into seed, up to seed, whatever the portions we are going to cover now, these processes are called, or these phases or events are called post fertilization events. So let us discuss one by one what are those events. So as the as the pollen grain fertilizes our central cell and zygote, okay, after that here primary endosperm nucleus is formed around the central cell and the zygote is formed from the ovum or egg. Then the synergid and antipodals, they have no work. Okay, so then they will start degenerating, and as I told you, your cell division is of two types, one is meiotic, one is mitotic. So here what happens after the development of zygote, whatever the development up to our embryo, all these cell divisions are what? They are mitotic cell division, because here what happens, no involvement of gamete is there, and we know in reductional division or meiotic division there is involvement of gamete, where from the mother cell the daughter cells receive only half set of chromosome. But here you will, if you will check this, these are except primary endosperm nucleus, zygote is 2n. Okay, this primary endosperm nucleus is 3n, because here we will find three nucleus. As we have discussed yesterday, two nucleus were already there as polar nuclei, and another nucleus will be received from the male gamete. Okay, then it is developed into zygote. Then the zygote will divide mitotically, and we know any living organism, when it grows in size and its organs or parts are developing, that is only by cell division. The cell number, the numbers of cell increase, and the size and the materials inside the cell, they also develop. Okay, so with that only a thing grows. As we are baby, we are now growing, our size has grown, because in our body new cells are formed, cells formed to make tissue, from tissue organ and organ system. Similarly here, the zygote will divide mitotically, and it develops into the first phase of embryo, which is called globular embryo, which looks somewhat like this. Okay, and here a group of cells develop which helps in attaching to other cells, which are called suspensor cell, and that suspensor cell, it will again remain persistent in the next phase, that is called heart shaped embryo. It is called because it looks somewhat little bit like heart. Okay, so it is heart shaped embryo, and here the development of cotyledons will also take place, as integuments already there in the cell, but cotyledons were not there. What are those cotyledons? Cotyledons, they contain the nutrient part in the seed. Okay, so cotyledons also start developing, and finally this is the structure of a matured dicot embryo. Okay, why have I written dicot? Because you can see here two cotyledons are there, and I know dicotyledonous plant, they have two cotyledon, and cotyledon are the half portion of a, in case of dicot embryo, half portion of a seed is dicot, but earlier they are present in the embryo. Okay, so let us discuss now about the structure of dicot embryo, and this diagram, this is the one diagram, and another diagram is also given in your book. They both are important from board exam point of view. I have already written here diagram of Mage TS of dicot embryo. TS of dicot embryo, it may directly given to draw, or the diagram is given, or it will be asked to label this part and write its function. Like cotyledon, here it is given and it will be given in A. Describe the function of A. Such questions may come in, you can expect in your board exam. Okay, now let us discuss. So I have already written the features of dicot and monocot embryo. This difference can also come, therefore I have written here. Then we are going to compare them. First, let me just make you understand which part is what. So you can see the dicot embryo poses only okay. I think it is some mistake. I think this is the This is monocot embryo. Okay, this is monocot embryo. Okay, so this is dicot embryo. Okay, so you can see here. Uh, First, let us discuss the dicot embryo. It is dicot embryo named so because it has two cotyledons. Okay, and the portion below the embryonal axis. What is embryonal axis? If I will divide this embryo into vertically or horizontally, we can imaginary line can pass through it horizontally or vertically. Here we can take it horizontally because the diagram I have made vertically. So that imaginary axis will be called embryonal axis. Okay, and the portion below the embryonal axis, this entire area is called epicotyle, and the epicotyle, it you can see this portion below, this portion is called epicotyle, and this epicotyle, the terminal part is called plumule. Okay, you can see here it ends with plumule, and plumule already know it will be also present in your seed. Plumule is that part which later on develops into what, root or shoot. It develops into shoot. So whatever the materials or cells that will be developed into shoot, that is present inside this shoot region. Okay, and the cylindrical portion below the cotyledon, okay, just below the cotyledon, it is radical. So here it is below written because I have drawn it in vertical, inverted. In book you can see it is given like this, just reverse the picture. Okay, so therefore in book it is written the portion below your epicotyle, this is epicotyle, and below epicotyle radical is there, and inside the radical your root cap is stored. This root cap has the cells which will germinate into root when the embryo is converted into a seed. I hope you understood. Okay, so these are the three main points regarding our what, transversation of dicot embryo. One point I missed, that why it is called dicot embryo, because it has two cotyledons. Okay, you already know these things about cotyledon. This is dicot embryo. Okay, fine. So now see, let us now come to monocot embryo. Monocot embryo is a little bit easier to remember. Whatever the features you can remember, but the diagram is a little bit clumsy and not hard, I will not tell, but it mostly does not come in your exam. Rather, the structure of your maize seed, okay, the structure of maize seed there in your book, and you have also drawn that diagram in 11th class also, so I'm not going to cover that one. But in maize seed you must remember the overall portion of the seed, it is covered by cotyledon because they are more nutritious. Okay, the monocot seeds, they are more nutritious. Therefore, most of the grain like our wheat, like rice, they all are what? They all are monocot in nature. Okay, so let us discuss the characteristic of monocot embryo. So you can see here this region is called epiblast. Okay, and here only one cotyledon is there which is called scutellum, and this will also remain persistent in our what, seed, which portion it will rather will develop more. Here it is not that much developed. Okay, in seed it will be present in more part. In the maize diagram you can see there. Okay, the lower end of embryonal axis terminates at radical. Okay, so where is the embryonal axis? Here, if I will pass a imaginary line, that will be embryonal axis. This is the upper half, this is the lower half. Okay, so the lower part consists of what? The root cap. This region is called root cap. Okay, and the root cap ends in a sheath-like structure. Means the root cap and radical, they are again covered by this portion which is called coleorhiza. So sometimes what is coleorhiza? What is its function? It may come. So you can write coleorhiza is the terminal part of monocot embryo which covers or it acts as a sheath for root cap. Okay, that protects that contains the radical, and all of you know radical has the cells that will be germinating into roots. Okay, and last part is epicotyle has leaflet like structures. Here radical has root cells. Similarly here the epicotyle, this region, it has, okay, the epicotyle has leaflet like structure, this one. Okay, and that region is enclosed, that leaflet like structure is enclosed within coleoptile. So we can also call it plumule like structure. Okay, but this generally mainly consists, or this generally mainly it covers the coleoptile region inside which the leaflets are produced. I hope all of you understood what are the different phases of your embryo. Okay, different phases of uh, from zygote to embryo, and what are the differences between dicot embryo and monocot embryo. All of you understood or not? Okay. So another thing here we have discussed only about zygote. Zygote has developed into dicot embryo or monocot embryo. But what about the endosperm? Here I have not mentioned that. So I am going to rub it, and we now will discuss what is the fate of endosperm, what the endosperm will become after, or how the development of endosperm will take place. So there also we will find two types of endosperm, which just I have given some glimpse in the previous class during triple fusion. Okay, right? So we have discussed about zygote only, but we have not discussed about endosperm. So who can tell me from which structure endosperm is developed? From which structure? Yes. So let us first not start with endosperm. We will write first polar nuclei. Okay. So polar nuclei, when the one male gamete, it is already 2n. When another male gamete will, or male nucleus of one gamete will fuse with it, it is converted into a triploid cell. Okay. I am writing 3n means triploid, 2n means haploid, and n means, sorry, 2n means diploid and n means haploid. Okay. So that triple cell is called, the nucleus will be called, here I have seen, I have written PEN. PEN stands for primary endosperm nucleus. Okay, primary endosperm nucleus. And similarly the polar cell, inside the polar nuclei are present where? Inside the central cell. So the cell is central cell and the nuclei are polar nuclei. If polar nuclei converts into primary endosperm nucleus, then the cell will be converted into what? The cell will be converted into primary endosperm what? Cell. Okay. And lastly, again it will divide mitotically and it is converted into a bunch or mass of cell which is known as endosperm. Okay, which contains millions of or thousands of cell within it, or thousands of nuclei within it. Okay, so endosperm is the structure, okay, which is the result of post fertilization, sorry, which is the result of post fertilization event that is developed from our what? Central cell. Okay, and it is very important. What is its function? Okay, endosperm, it contains the nutrients necessary for what? Necessary for development of seed. Okay, development of seed. So here you can see it started with zygote and ended with a large structure of embryo. So from where all these nutrients they are getting for all these entire cell division? Here one, not a cell, single cell is there, numbers of cells are there. So from where they are getting these nutrients? These nutrients are obtained from endosperm. Okay. So endosperm mostly it is not found in our seeds. Okay. During this post-fertilization event or embryonic or development of different phases of embryo, this endosperm provides nutrients, so it itself gets completely used up. Okay, it is rarely found in any type of seed. So on the basis of that, we have divided the seeds into two types. I will come into that section while we will discuss about seeds. But before that, let us discuss what are the two different type of endosperm are there. Okay, so first one is our cellular endosperm. Okay, cellular endosperm. Another is our pre nuclear endosperm. So these are the two type of endosperm which are found in nature. Okay, cellular means here the endosperm is found in the number of cells. Endosperm is found in form of cell. The best example is what? White kernel of coconut. Yesterday I had told you white kernel of coconut. You can expect this question in your MCQs. Okay. Second, what is free nuclear endosperm? Here many nuclei can be found in form of endosperm, and it is also found in the same fruit, that is best example is our coconut. Okay, the liquid part or the water part of coconut is what? Our prenuclear endosperm. Okay, so here we can get two types of question. One is your MCQ, another you can get what case based question. Okay, embryonic development it may be given, about endosperm paragraph it may be given, and question may be asked from this part. Okay, and another question may be asked about albuminous and ex-albuminous. I'll come to that later on. So I hope this is the entire post fertilization event. Okay, and after the this so all the development are done, means endosperm is developed, endosperm is used, and your all these zygote is developed into embryo. Then what will happen? The rest of the part of the flower, petals, sepals, they will be first threaded. Okay, then the male part will be entirely threaded if it is a what if it would have been a bisexual flower. Only what happens, the ovary part remains persistent. Okay, because as we know, inside the ovary what is there? Inside the ovary our embryo sac is there, and this embryo sac is now developed into embryo. Okay, and the ovary inside which the embryo is present, obviously that will develop into something. So ovary has flesh like structure which is known as thalamus part. Okay, and therefore the ovary will develop into fruit. Okay, so if I will write which part of plant converts into what after post fertilization, then I will have to make a small table. Okay, which already of all of you know from your lower classes, but just for your revision I will tell you. Okay, so after all the rest of the part of your flower they have degenerated, only this ovary or thalamus portion is there. You will see the ovary will be developed into fruit after post fertilization event. Ovary is development of fruit, or you can say these are the result of your post fertilization event. Okay, the ovule or embryo sac is developed into what? Our seed. Inside this ovule only the development of embryo takes place, and finally the embryo converts into seed. Then our integuments, the integuments of ovule or microsporangium, megasporangium, they develop into seed coat, the outer layer of seed. Okay, so these are some important things which can be found in your ovary, which is developed to fruit. Okay, and ovule is developed to seed, integument development to seed coat. Okay, so this is all about your post fertilization event. Now we will discuss about seed. Okay, our next topic is seed. So just wait for a moment. Okay, so let us move to our next topic, that is seed. Okay, so here you can see seed. Here I have already told the structure of maize is there. When we generally discuss the structure of seed, we take the example of a dicot seed, like your you have seen that groundnut. Okay, or you can see that gram. So such type of seed we take into example. If I will draw it, you can book in book also it is given like this. Such type of seed we are found. Okay, and here this part is called what? This part is called radicle, which later on grows into root. Okay, this part, sorry, this part is plumule. This part is plumule, which develops into shoot. This part is called radicle, which develops into root. And all these things are also present in our dicot embryo also. Okay, and this one is the embryonal axis. You can see the embryonal axis which is dividing it into two different portions. It is that embryonal axis which remains present in our seed. Okay, so that seed line or embryonal axis is still there. If I will cut the seed from this part, you can open the seed and you can find some structure like this. Okay, two different parts of the seed. Okay, this is your radicle, this is your plumule. Sorry, this is your plumule, this is radicle. So what are these two different parts are called? These two different parts are called cotyledons. Okay, these two parts are called cotyledons. So what is the function of cotyledon? This cotyledon is the source of nutrition during what? Seed germination. So this is the source of nutrition during seed germination. Okay, And mostly your cotyledon in dicot plants like your legumes. Okay, legumes, they are mostly what? They are proteinous in nature. Therefore, you can see whatever the dal pulses we used to take, they are highly rich in protein. But when we talk about maize, maize has only single cotyledon and it is much more rich in starch and other carbohydrate. Okay, so all the grains or cereal crops, they are single cotyledonous and they have more nutrition as compared to our pulses. But pulses have more proteins. Okay, so this is all about your seed. Then what happens? How the seed develops? We have now seen, we are reading about reproduction. That means one plant, how it develops into another plant. We have not got the plant till now. So the next topic, how we will get the plant from seed? By seed germination. Okay, seed germination. Another thing I forgot to tell you, that the integument, okay, integument as you know converts into seed coat. So this outer part of seed is called seed coat. Okay, seed coat. So how seed germinates? Okay, what happens? Seed, okay, waits for correct temperature or correct, okay, correct temperature or correct moisture. Not wait, needs, need will be the more appropriate term. So a seed needs more correct temperature or correct moisture for germination. Okay, for germination it needs. So what is seed germination? We will come to seed germination later on. Before that, we must know, unless and until the the favorable condition does not come, you have read about it in 10th also. Okay, favorable time doesn't come, it will not germinate and suspend all metabolic activity. Okay, it suspends all its metabolic activity. So what will happen? It will be inactive. This phase of inactivation, this phase of inactivation is called what? Is seed dormancy. Yes. What is the meaning of dormant? When you are not going, moving out of your house. Okay, you are living inside your house like in corona phase. You are doing nothing. So I can tell you are socially dormant. Okay, you are socially not active. So here the seed, it falls in the ground and in the ground itself near the beside the layer of soil, it remains inactive. This phase is called seed dormancy. Seed dormancy. Okay, so how it germinates then? So as I told, for germination it needs correct temperature or correct moisture. Okay, so where the temperature or moisture, it will how it will enter into the seed? See, here a small hole remains persistent in the seed, that was persistent in your embryo sac, not only embryo sac but also in your ovule. All of us know, when I taught you regarding the structure of an anatropous ovule, there we saw that the upper part was chalaza end and the lower part was micropylar end. Okay, that micropylar end, now it is also remain persistent in the seed, which are present in microsporangium or ovule. Okay, that is found in embryo sac and also here. So that micropyle hole or pore, it still can be found in the seed. And what is its function here? Okay, here through micropyle moisture or air can enter the seed. So you know, seed has many cells, they also need oxygen, they also need to breathe. So the site of breathing for them is micropyle. Another important fact you must know, that how much moisture they required for germination. Remember, they required 50 to 60. Okay, they required 50 to 60 percentage of moisture for germination. Is it there in your book or not? This part. Okay, this question mostly asked, this factual questions, don't think that this will not come. Therefore, I have written it red color. Okay, it will come in MCQ in board exam. Directly it may be asked. Okay. So remember, 50 to 60% of moisture required for germination. Okay, for germination. So these are some points which are required for exam. Okay, then after the perfect time will come, then what will happen? Seed becomes active, and after that whatever happens you all know. Okay, this plumule will develop into shoot, and the radicle will develop into root, and all those nutrients required for this process, it is stored within the what? Your cotyledons. So I am not going to cover that part. You will get it in your notes. Okay, seed germination of this part you have already read in your what? 10th class. Okay, so we'll now move to our next topic, that is fruit. So here in the fruit, seed, another thing is there. For that I will have to rub here. Seed is not over. A very small portion I have left. Okay, that I have to rub here, and then we'll discuss. Then we'll move to fruit. You can see here. Here I have written two terms. अभी कुछ देर पहले मैंने आपको बोला था that दो तरह के seed होते हैं depending upon the when I was teaching you endosperm. There I told you two types of seed are there on the basis of whether endosperm is completely used or partially used. Okay, so you can see here this is the albuminous seed. Okay, the albuminous seed, they are completely used up. Okay, what are albuminous seed? Here the endosperm is completely is it complete? Sorry, I did a mistake. Therefore I had some doubt, I paused there. Okay, in albuminous seed the endosperm is persistent, not used. Persistent means they are found to be present. Okay, so they are called albuminous seed. Wheat, maize, castor and barley, some of their example. Okay, therefore they are highly nutritious. Okay, because endosperm is found in them. But in eggs or non-albuminous seed like your pea plant, motora, and groundnut, your mungfali, there will find endosperm are completely used. So those seeds are not that much rich in carbohydrate, but they are rich in good fat, good amount of fat and protein. Okay. Then another term is there, perisperm. So I'm not going to write in detail about that. You can get 2 mark question. So you can write according to that. Next is your perisperm. Okay. What is perisperm? That I will discuss with you. See, I told you while teaching the structure of your anatropous ovule or megasporangium, that the suppose this is the embryo sac. Okay, this embryo sac, it is covered by a special type of tissue, a mass of cell of tissue which is called nucellus. Do you have remembered or not? And above that different integument was there. Okay, hilum was there, funicule was attached to, like the structure was there. This bunch of cell which is called nucellus, in some seeds this nucellus is persistent. Okay, so nucellus is persistent in some seed in in the form of what? In the form of perisperm. Okay, it can be still found in form of perisperm. Such seeds are called what? Such seeds are called perispermic seeds. Such seeds are perispermic seeds. So you can sometimes it is given persistent, or in some books you will find the word reminiscent. Reminiscent means what? कुछ बच किसी portion, very small portion of something if it is left, that is called reminiscent. Okay, so that word you can also find in some books. Students does not understand and they make some mistakes. Okay, they get confused between perispermic seed and this albuminous or endospermic seed. They are both the same, so student gets confused between these two. And ex-albuminous, non-albuminous, they are also called non-endospermic. Also, they can be called. Okay, so this is all about our seed. Now let us discuss our next topic, that is fruit, which is a very small topic, but you can expect some MCQ or single mark question from it. Okay. So here I have already written from a diagram point of view, your fruit is a little bit important. The diagram of apple. Okay, that cut apple, which is the two pieces have been shown. It is given in your book. That is already been asked in your board exam question. Okay, the diagram was given, and the for labeling it was asked, and for the maize seed it was asked completely to draw. And in another year, the diagram of maize seed was there, and they asked to what label it different part and also write its function. So you have to cover these two topics: diagram of maize and diagram of apple. Okay, that figure 1.15 it is there. So just I will discuss what are the different parts of fruit. When we are discuss about fruit, so which part of the fruit develops into which part of the embryo develops into fruit? Which part of our flower? The ovary. It is developed from ovary. Okay, but from which part it is actually developed? Okay, on that basis, fruit can be of two types. Here I have written one is true, one is false. So what are true for fruit? True means any fruit, okay, developed developed from thalamus, okay, developed from thalamus of ovary. It is called true fruit. Example I have written your mango, okay, orange. They are example of true fruit. What is false fruit? False means except the thalamus of ovary, any part it is developed, then it is called false fruit. Okay, fruit developed from any other part of ovary. Okay, so this is called false fruit. So this much question can be asked. Then again, there are types is there. What are the different types of true fruit? What are different types of false fruits? These things, if you want, I can make some another session for it. Okay, but for board exam point of view, that is not important here. But another type of fruit is there, which is called parthenocarpic fruit. Parthenocarpic fruit. What are they? Here, two important points you have to remember about them. One is first, such fruit, such fruits, such fruits don't have seed. There is a reason why they don't have seed, because these fruits develop without fertilization. So they can be developed without fertilization. Got my point or not? So here you can get a question from assertion reasoning, parthenocarpic fruit, and this process is called parthenogenesis. This phenomenon is called parthenogenesis. Both of you are clear? Example? Can anyone give me example? One example Swastik give me, one example Ananta you give. Okay, banana. Another example Swastik? Strawberry. Okay, nowadays by artificial method many seeds, parthenocarpic seeds you can find. Okay, your what? In your orange also you can find. In orange also, nowadays some oranges are found in market which have no, not only orange, lemon also found having no seeds. Okay, so parthenogenesis it has what? Pear also you can get. So parthenogenesis what? It has very demand, or it has very, what is its application? Application in field of agriculture. Okay, in field of agriculture. As you know, parthenogenesis is a asexual mode of reproduction because here no fertilization is there. So what happens? The the yield is much higher. Yield is much higher. At the same time, you can produce higher number of fruits. Okay, and cost is also low. In such case, cost is low, because normally when you have to wait for the fruit, you have to wait for the perfect temperature, you have to wait for the perfect season. But this nowadays you can see all of them are found in any season. Okay, nowadays mango, which is a summer jet crop, you can find it in January also. Okay, so that is possible only due to this parthenocarpy. And in lab nowadays, such researches are also going on. So this is very much important step for us also, and we have artificially, okay, exploited it for our own benefit, right? So now let us discuss the last portion of this chapter. So with this, our seed and fruit is over, and the last topic we will discuss about, that is apomixis. Okay. So just now I told you, just like your fruit which is developed without fertilization is parthenogenesis or parthenocarpic fruit. If any seed is developed, yes, seed can also be developed without fertilization. That is called apomixis. And today, whatever I taught you, the most important question is apomixis regarding your board exam, because it is mostly repeated. It is very small topic, but it is mostly repeated, and at the end of this class, I will tell you what question may come from it. Okay, what questions may come from our apomixis topic that we will discuss. Because here I have written only the topic. Okay, you can express, you can what expect the questions directly from one to these three topics. But apomixis, there are certain specific questions. Okay, different types of question may come, that I will discuss, which we have already seen in last 4 years or 5 years after Corona. Okay, so see, first question, let us understand what is apomixis. Apomixis means development of seed without fertilization. Development of seed without fertilization. So how the seed is produced actually? Actually, what happens? It mimics which reproduction? Sexual or asexual reproduction? Anant. It mimics the asexual reproduction, because here no involvement of gamete is there and no fertilization occurs. So no fusion of gamete. Then seed is formed. Okay. And in which type of plant it is mostly occurs? It is mostly occurs in the family of grass, Astraceae. Okay, it is found in mostly the family of Astraceae, which contains the variety of wild grass. Variety of wild grass can be found in this type of plant, this type of family. Okay, Astraceae family. It may come in your exam, so keep it in mind. So this is the definition, or you can get some assertion reasoning based question from this portion. Okay, AR question means assertion reasoning. But what is the important question I am talking about? The question is, why the demand of apomictic seed is increasing in the field of what? Hybrid seed industries. Hybrid seed industry. This is the question which you can mostly found. Or write the significance of what? Apomictic seeds. So first I will tell you what are the problems, or question may come in broken portion also. Okay, question may come like this. I will write the question only and I will discuss with you. You will get the detailed answer in the notes. Okay. Another question we get in AB form, that what are the problems faced by farmers while using hybrid seeds. Then such type of, it is the indirect question of apomixis. The question may be asked, how they overcome this problem, how they overcome this problem. These two questions are, this can be also asked in many different format also, but if you know the answer of these two questions, you can find any type of question, you will find the answer related to apomixis. First is the definition. Now this portion let us discuss. So what is the phases that are produced in hybridization industries when the farmers produce naturally, when the farmers produce any hybrid seeds naturally, what are the problems? Let us discuss. So what is hybridization? Before discussing into it, I will want to pay your attention on the topic hybridization. Like we have discussed yesterday in artificial hybridization. Hybridization is a process when we mix or collaborate the different breed seeds or different breed genes with each other. Then the next new gene or the new type of seed we get, that is called hybrid seed. In case of plant we seed, in case of animal you will call hybrid animal, cross breeding, and they are mostly result of cross breeding or cross pollination. Okay, so in hybrid seed, suppose for example, we know that basmati rice, it is a what indigenously produced hybrid rice. Okay, our forefathers or our farmers of India, they have developed such many varieties of rice naturally. Okay, by hybridization, but they have to face a problem here. What? Since we know that if it is a hybrid seed, that means it has some character or some what significance of its own. Like we know that basmati rice, it is very tasty and it has a very good aroma. Okay, मतलब आपके घर में जब भी बिरयानी या फिर फ्राइड राइस हो रहा है तो अगर बासमती चावल का यूज करें घर में पूरा महक चारों ओर फैल जाता है. सो दैट इज द स्पेशलिटी. तो यूएस ने क्या किया, उसको अपने नाम पे कर दिया था, फिर इंडिया ने बहुत इधर उधर का हाथ पैर मार के अपने नाम पे किया पेटेंट. ठीक है. तो लेकिन दिक्कत एक यहाँ पे है कि जब कभी भी हम लोग हाइब्रिड राइस या हाइब्रिड सीड्स वी प्रोड्यूस द सेम कैरेक्टर which the hybrid seed has, it will be transferred to its next generation or not, that is not sure, because hybrid seeds are produced by sexual reproduction. Cross breeding is sexual reproduction. So again, new variety can be produced in the next generation. That is a very problem which farmers have to face. So they have to do it very carefully, and it is also cost expensive. So therefore hybrid your basmati rice is costly. You can see any hybrid thing in market, whether it is fruit, whether it is flower, or whether it is our cow, their product is more costly, because the same quality to be maintained in the next generation, it is not that much easy. So it is the problem. This problem is faced by the farmer. Okay, and you have to produce that hybrid seeds generation over generation. That is a problem. Okay, बार बार आपको क्रॉस करना पड़ेगा. So how, now the question is how you can overcome this problem. So the solution is apomixis. Research is now going on. So in some of the plants they have already succeeded, but researches are still going on. If we will able to produce the hybrid seeds apomictically, means just by asexual reproduction or without fertilization, then what happens? We know that in asexual reproduction there is no variation. In hybridization there is variation in sexual reproduction. Means if I will produce the basmati rice asexually or apomictically, the same character will be retained in its next generation also, because no sexual reproduction is involved. Okay, because there is no sexual reproduction is involved. Okay, so if we will able to produce the hybrid seeds apomictically, we can get the same quality and same characters generations over generation, and it will reduce the energy, time, and also it will be cost effective. So these are some of the significance of your apomictic seed which I told you. Okay, and why the demand is increasing, that is also the same reason. Both of you understood? Okay, so this is a very important question. This is the most important topic what we have discussed today regarding the today, and these are the some topics you can note it down. Okay, so thank you for joining, and in the next class we will start the new chapter, that is principle of inheritance. Okay, then we'll go for molecular biology, then we'll again come back to our reproduction unit. Okay, so any doubts in today's class? Polyembryony, that topic I left. I think that I forgot to mention polyembryony. That should have been discussed under that seed only. Polyembryony means what? When I told that a single embryo is produced within the same fruit, or by single fertilization, single embryo is produced. Okay, when multiple embryo are produced within a produced in a single fertilization, are produced in a single fertilization, that is called it is called polyembryony. Understood? Can you give me one example of it? Polyembryony. All? Yes, tell. Yes, mango can be there. And all citrus fruit, all citrus fruit. So where you are finding more than one seed, seed kahan se aaya? Seed is coming from embryo. So there are multiple embryos are there. So it is called, they are example of polyembryony. Your guava, mango, fruit, what all citrus fruits, lembu jatiyo jitna bhi aapka nimbu related jitna bhi aapka hai, they all are, so this is called polyembryony. I forgot to mention. Okay, so with this I would like to end this class. Thank you and good night. Okay, we'll meet in the next session. Thank you, good night.